everyone, Professor Davis here, and the last body system group that we're going to discuss here in microbiology is going to be the nervous system. So let's dive into the diseases here of the nervous system. So a little introduction, here are some terms, things like meningitis. Meningitis is the inflammation of the meninges. The meninges are the special covering that covers the brain and the spinal cord for protection. We also have encephalitis, which is inflammation of the brain tissue itself. Now, what kind of therapies do we use for nervous system infections? Well, we can use chemotherapeutic drugs, but they have to be lipid soluble. This is because there is a blood-brain barrier that is created, and we have to be able to penetrate through that. They have to be able to cross that barrier. Now, when we look at this, meningitis, some general symptoms of meningitis include fever, headache, and a stiff neck. The stiff neck is actually one of the cardinal signs of meningitis. It can progress ultimately to convulsions and a coma. The individual, even with recovery, may suffer from neurological damage. Now, viral meningitis tends to be more common, and it has milder effects on the patient. Bacterial meningitis normally can cause death from release of endotoxins. These are normally going to be caused by gram-negative microbes, and it results in shock and inflammation in the system. Vaccines are available for some of the strains and they have reduced incidence for us, like Streptococcus pneumoniae, Haemophilus influenza type B or Hib, and Neisseria meningitidis. Now, when we're talking about identifying different things that might be hurting the nervous system, we may have to do a cerebral spinal fluid diagnostic test. These cerebral spinal fluid diagnostic tests are going to be where they use a serological test followed by culturing of whatever might be present. So when we look at this, the serology has to be done under an hour, and then the culture is going to be at 24 hours preliminary and then 72 hours for the final results. And it's important to process the specimen quickly. Pathogens do not survive well once outside of the body. Now, what we would notice here is when they collect the cerebral spinal fluid from this test, they normally will collect 3 tubes. The first tube is for chemical analysis, looking at the glucose, protein, and other structures within the cerebral spinal fluid. The second is the hematological test, where they're looking specifically for blood and white blood cells in there. And then tube 3 would be the microbiology test. There are some rapid tests that are available, but a negative test does not exclude possibility of bacterial meningitis. So further tests would have to be done with the plates. So let's talk a little bit about some of the bacterial infections of the nervous system. So of course we have bacterial meningitis. These guys are normally going to have capsules and toxins, and those are the 3 we talked about a little bit earlier. We also have Listeria monocytogenes, Clostridium tetani, Clostridium botulinum, and Mycobacterium leprae. So let's talk about each of these in a little more detail. So Streptococcus pneumoniae, we've learned about previously. It is normally a nasopharyngeal normal flora. It's in your nose, and you're normally a carrier of it. And it can cause pneumonia and even middle ear infections. The problem is that it is the leading cause of meningitis today. So complications can occur from this. Now, one reason why it's the most leading cause is because there are vaccines against the other causes, so they are going down, and this one does not have that. The mortality rate is higher in children at 30% and the elderly at 80%. There are vaccines available, but it's not as good as the ones that we are seeing on other cases. So with Streptococcus pneumoniae, remember that it is gram-positive. It has more of a lancelet shape, so it's diplococci. It is also alpha hemolytic. It doesn't destroy the red blood cells like the beta hemolytic do. It is encapsulated. And when we look at this, its strep pneumoniae is going to be positive for the Oppigen test, whereas other alpha strep that are not pneumoniae are not going to be affected. The next one is the Haemophilus influenzae. This one is going to be a normal throat flora as well. We a lot of times are, again, carriers here. It does cause pneumonia and inner ear infection or middle ear infections. We see that this H. influenzae type B is also known as Hib, and it can cause meningitis as well. Now, historically, it was the leading cause of meningitis in children, but today, again, it is on decline due to effective vaccines that are available. So if we take a closer look, these have been seen before. Remember that they are tiny gram-negative coccobacillus. We also see that they are fastidious, they are picky, and they are blood-loving bacteria. They need both the NAD and the hymin, which are going to be factors V and X, growth factors. They need both. When we also look at Haemophilus influenzae, it will grow on the chocolate agar, the cap agar. Remember, those are laked blood cells, and this is why they grow, because it has that X and V structures present. This one also does smell like dirty socks as it's growing on the plates. Haemophilus also has satelliting around beta-hemolytic Staphylococcus aureus when it does grow on blood agar. And again, these are all things that we have already looked at before. The next one is the Neisseria meningitidis. This one is also a nasopharyngeal normal flora. So we are, again, carriers, about 40% of the population. Symptoms are due to the endotoxin production. So quick onset of a sore throat, then it causes septicemia, and then the meningitis. We do see that the at-risk populations are children under 2, college students, and historically the military. Now, wait a minute. Where does it jump to? Why is it affecting college students and the military? Well, this has to deal with the close contact with the use of dorms. The fact that they're living in closed quarters with each other, that is going to be where they have the spread of this particular type of meningitis. Again, vaccines are available for some of the serotypes, but not the childhood types. So this does create a potential issue. When we look at this group, you do remember that is gram-negative diplococci. They look like little kidney beans. We do see that they can also grow on blood agar. They are oxidase positive. They're also aerobic, meaning they like oxygen. We see that if we use the CTA sugar test, the cysteine tryptic agar, they are positive for fermenting in glucose and maltose, but they don't really like lactose and sucrose sugars. The next one we want to look at is the Listeria monocytogenes. This is a type of bacteria that is gram-positive, and it is a bacillus species. It can actually thrive in cooler temperatures like the refrigerator. So this causes it to be a bigger issue. It causes neurological disorders and stillbirths, specifically in pregnant women. This is a problem. They are at the high risk. So immunocompromised adults, where they can get foodborne illness, sepsis, and meningitis, or even fetuses or newborns where it crosses the placenta and it can actually cause stillborns, which is the abortion, or meningitis. When we look at Listeria monocytogenes, it's going to be found in certain food products. This is going to be coming from reservoir of animals, and it does get transmitted through the fecal-oral route. This is due to poorly pasteurized dairy products or foods in contact with raw beef, pork, or poultry foods, and raw vegetables. Guys, this is why it's so important that you wash your vegetables before you eat them, especially if you're going to eat them raw. And also, lunch meat is another one that causes an issue, and so it's important to heat up your lunch meat before you eat it. This is also another reason why you're not allowed to eat sushi while you're pregnant. This can cause mild symptoms with the mother as she's pregnant, but it could be highly fatal to the baby. The next one is Clostridium tetani. Clostridium tetani is also known as tetanus or lockjaw. Deep wounds with spore-contaminated soil is what is going to be the issue. Symptoms from potent neurotoxins, known as exotoxins, are going to enter into the central nervous system through your peripheral nerves. This creates really bad muscle spasms. And then a lot of times the patient needs supportive respiratory care because the diaphragm will contract, which allows them to breathe in, but it cannot relax to allow them to breathe out. There is a toxoid vaccine available for Clostridium tetani. It's the DTaP. It does help against diphtheria and pertussis as well, but it stimulates the antibody production that neutralizes the toxins. You should get a booster about every 10 years. Now, if you are more prone to being exposed to this, you may need to get it more often. Now, treatment for tetanus includes antitoxins called TIG, TIG. These are borrowed antibodies, and then we also need to do wound debridement to clean out all the bacteria in the wound. We then have, when we look here at Clostridium tetani, it is gram-positive anaerobic bacillus. They kind of look like boxcars. They do have these terminal endospores that are present. Muscle spasms are going to be due to the neurotoxins, and this is contraction without relaxation. So that's a big deal. It's contraction without relaxation. The next one is the Clostridium botulinum. This one is also known as botulism, and it can cause food poisoning. A lot of times we see individuals being exposed due to poor canning techniques. These poor canning techniques promote the growth of this anaerobic bacteria. They also produce a neurotoxin, but it's different than the tetanus. The tetanus causes contraction without relaxation. This one causes relaxation and no contraction. So it paralyzes the muscles. So when we look here, it interferes with the neurotransmitter, specifically acetylcholine. Endospores are going to be heat stable, and if you ingest them, they can cause either no harm for the most part, but the exotoxins are heat liable. They can be destroyed if you heat them up. Botulism is going to also have antitoxins available for treatment. Now, Clostridium botulinum is a gram-positive anaerobic bacillus. It does have terminal endospores as well. But guys, one of the reasons why you're not supposed to feed babies honey before the age of 1 is due to botulism. This can cause infant botulism, and this is because the endospores can multiply in the intestines and produce the neurotoxins when they're present in the honey. As adults or over the age of 1, we don't have issues with this. We can also see wound botulism that could be present, and then guys, the toxin that the bacteria Clostridium botulinum makes is actually what's used in Botox treatments. It has cosmetic usage, but also it can also reduce perspiration, so like sweating. And we also see it can be used sometimes for migraine treatments. The next bacteria is the Mycobacterium leprae. This is also known as leprosy or Hansen's disease. And you're probably like, leprosy? I've only really heard about that in the Bible. That's true, but it's also present even today. We see that it grows in the peripheral nervous system, which does result in nerve damage. And the reason it likes the peripheral nervous system is it prefers cooler temperatures, around 30 degrees Celsius. So this is why it causes issues with the fingers, the toes, like with the nose, that kind of thing first. What these guys do is they secrete organisms from the nasal mucosa. It's not very contagious, and you actually need really close contact for prolonged periods in order for you to become infected with it. Now, you're probably wondering why we have an armadillo on this picture or on this slide. This particular type of bacteria is very difficult to grow in the lab on media. And so tissue cultures are normally used most often for this, and armadillos are really good with this in their fat pads of nude mice because it's the cooler temperature where they can actually grow better. We also see, because it's a mycobacterium, gram staining is not helpful, and so we have to use the AFB positive stains here. And another thing that takes this a long time for it to grow in the lab is this bacteria reproduces very slowly, and it can take 12 days for it to actually replicate. This bacteria was first isolated in the 1870s. When we look at this Mycobacteria leprae, you can see that there is what we call the tuberculoid form. The tuberculoid form is shown on the skin here. We also see that they will have to live in what we call leper colonies. This is where individuals who actually have been diagnosed with leprosy are kind of outcasts because they don't want to spread this disease. When they do come in contact with other individuals, they normally would have to yell like unclean within those groups in order for people to know that they're there. We see that a lot of times they will actually lose their fingers, toes, and different structures from this disease because this is a cell-mediated immunity response which tries to get rid of the areas infected with the bacteria. There is a BCG vaccine that is available for tuberculosis, which could also use to help for some protection against leprosy, because leprosy is also a mycobacterium as well. The next thing we want to look at are the viral diseases. These include polio, rabies, and arboviruses. Arboviruses are things like arthropod-borne encephalitis. That's where the arbro comes from. These include things like East and West equine encephalitis, West Nile virus, and Zika. So let's talk about poliomyelitis first. This is caused by the polio virus. It is an RNA virus. We do see that it is spread by ingestion of contaminated food or water. It does cause intestinal distress first. A lot of times patients that are first infected with polio will have a sore throat, fever, nausea. It only progresses to the central nervous system in only 1% of patients. So it's not very common that it will then go to the central nervous system and cause paralysis, but it can. Prevention is going to be inactivated virus vaccine. This is called Salk. We also see the annotated virus oral vaccine, which is Sabin. There are the two types. They were developed at different points. Most of the time, the Sabin one is the one that's used most often now. We do see that this particular virus does weaken the nervous system, specifically in the legs, and it causes the legs to be super weak. Now, this is a slow process progression, but it can occur. When we look at FDR, Roosevelt, he had polio. And he was able to stand and things like that, but he used a wheelchair a lot of times. If you'll notice that the Oval Office desk, one of the really famous pictures is when John F. Kennedy's son is playing underneath the desk with this kind of door opened. There's like a passageway. That door was added during FDR's presidency to hide his wheelchair so that he could sit in his wheelchair in the Oval Office with that closed and people wouldn't necessarily know. All right, and so that's part of when we look at polio. This is known as post-polio syndrome because it happens after the polio infection seems to have been taken care of. These are some of the complications that occur. The next one is the rabies virus. The rabies virus is Latin for rage or madness. That's what rabies actually means. This is going to be where it proliferates in the peripheral nervous system, but then spreads to the central nervous system. We have a really poor immunological response. Your immune system is not able to respond to this type of virus. This does cause a fatal type of encephalitis where the brain is going to be inflamed and cause death. The virus is transmitted by infected saliva through bites or wounds. A lot of times these bites are going to be from infected animals like dogs, fox, bats, things like that. Once it enters into the central nervous system, the immune system cannot fight it anymore. We see that in this case, it's called the Lissea virus, and it is bullet-shaped in its structure, and it can be actually given in transplanted tissues as well, specifically the cornea. They need to test for this before they transmit it to another patient. Now, when we look at rabies, it does cause some very unique symptoms. Because of the inflammation in the brain, the individual becomes scared of water. So they become very hydrophobic. They don't want to be around water. They won't drink water. And this is part of the reason, like due to the inflammation and the dehydration, it causes this disease to be fatal. The next one are the arboviruses. Remember, the arboviruses are going to be carried a lot of times by a mosquito, and they come from a reservoir of birds. These guys are going to ultimately infect a lot of times livestock in the sense of horses, as well as individuals who are outdoors a lot in the sense of camping or hiking. We do see there are different types, but all of them cause a type of encephalitis or inflammation of the brain tissue. West Nile was seen in 1999 for the first time in the United States, and we actually have West Nile cases constantly in Lee County where we live in New Mexico, but a lot of times they're still going to be mostly in horses, but we have seen some humans be infected as well. We do see that in the U.S., Zika has also started to become an issue, but more it's located in the kind of Caribbean areas. We see that chills, headache, fever, and it can progress to mental confusion and a coma if these guys are left untreated. And because they're viruses, we can't treat them with like antibiotics and all, but we can help support the person's immune system as they fight these diseases. The next group we want to look at are the fungal diseases. In this case, we're looking at Cryptococcus neoformans. This one right here is going to be stained with a negative stain, with that Indian ink stain, and it forms these really large capsules around it. We do see that we can grow it on differential media. The dark colonies are the Cryptococcus neoformans, whereas the light colonies are the C. albicans that we talked about earlier in certain fungal infections. These fungal diseases are spread a lot of times by pigeons. We see that a pigeon can produce 25 pounds of droppings per year. This creates a really big problem. The more pigeons we have, the more droppings there are, and the more chances that you could be infected by breathing in these spores that come from these organisms. We do see that people who are more commonly infected by this type of fungal infection are going to be the immunocompromised and the individuals specifically who have AIDS. It can cause meningitis as well. The next ones we want to look at are the protozoa diseases. The first one we see are the trypanosomas. These are going to be flagellated protozoa, so they have the tail. These guys cause what we call African sleeping sickness, and they are going to be passed or sent by the vector of the tsetse fly. Once these guys get into the central nervous system, they are very, very hard to treat. We see that the drug that's used to treat African sleeping sickness is actually very toxic, and it kills 5% of the patients who take it. So even though it may help cause survival from the trypanosome, you could still die from the treatment, okay, because it's very, very toxic. These flagellates are hard to fight due to their ability to create new protein covers. What happens, guys, is they hide in plain sight. They let your immune system see they're there and that they're an invader. And as your immune system prepares to destroy them, they change their shape. They put on a new coat, so they look different. But your immune system still notices they don't belong, but now what we made to fight the original proteins are not present, so we have to do something again. This causes the immune system to work extra hard over and over and over again, which makes the patient very tired, causes the patient to fall into a coma, and then ultimately results in death. The next one is the Naegleria amoeba. This amoeba invades through the nasal mucous membrane, and it moves to the brain and feeds on the nervous tissue. It ultimately can cause encephalitis as well. There is 100% mortality rate due to misdiagnosis of this particular amoeba. A lot of times, we don't even find it until the autopsy. They didn't even think that it was the culprit most of the time. So it gets misdiagnosed most of the time, causing death, and it is then found during the autopsy. The next one is the neurocysticercosis. This one is going to be a larval form that comes from the pork tapeworm, the Tinea solum, that we talked about previously. We see that it can then spread into the brain, and diagnosis is normally with radiological imaging like you see here in this picture. We do see with this that the diagnosis leads to treatment, which depends on the location, the number, the size, and the stage of these cisternae, these like pockets that are present. Okay, this ultimately will be determining what we will do with this. And this would be a worm infection because it is due to a flat worm. This is due to a tapeworm. The last disease we want to talk about are the puron diseases. Remember, purons are going to be misfolded proteins. The example here is Creutzfeldt-Jacobs disease and Kruar. When we look at this, the Creutzfeldt-Jacobs disease is the classical variant, and we do see that it could have some genetic type of component to it, whereas the Kruar is due to cannibals. And you're like, what? Cannibals? People still eat people? In some groups, especially isolated groups, they do have cannibalistic rituals when it comes to family members dying. They feel like they have to eat their brain and their heart in order for them to move on to the next life. This creates a problem because these purines are going to be a problem in the brain. They do not die from cooking it. And when you eat it and consume it, you then just infected yourself. So this creates a big issue. And then trying to convince them that there is something that we need to change with their religion, in a sense, is really hard to overcome with medical advice. So I know that this just went over briefly some of the examples. Remember, these are not all the diseases that are caused by bacteria, viruses, protozoa, fungus, all those organisms for the nervous system, but these are some. Make sure you are reading your book as well. And if you have any questions, please let me know.